Thank you, Jack, and thank you all for coming along. I'd particularly like to thank the director for his kind invitation, and the Tanner family for supporting this museum and this talk. So, as you've heard, I've had a bit of a career as a ping-pong ball crossing the Pacific back and forth and back and forth. And yet my accent hasn't yet converged on Californian, as you can tell. So if you're having trouble understanding me, just say stop talking in Australian, help, speak to me. And I'm happy to take questions as I go. Sort of late at night and a monologue tends to be a bit dreary, so do ask questions as I go along. So as Jack said, can you hear me okay up the back? Is this microphone thing working? Good. So as Jack said, my theme is really about the value of museums such as the one you're in. And often people see just the sort of superficial layer of the museum, which the public exhibits, but the real power and the enduring value of it is through that education that the public exhibits provide, but also the specimens themselves and the collections and the field notes and the photographs and all that stuff that goes with it, that, as I hope to convince you today, just gets more and more valuable in time, like a rare piece of art. And they enable us to answer questions that the original collectors didn't even think about or didn't imagine possible, with techniques that weren't on the radar at the time. So maintaining these specimens, maintaining the records, the databases and so forth, is really crucial going forwards in time. So the particular story I'm going to tell you about starts 100 years ago and will probably continue for the next 100 years. And it's about using these historical records, the specimens, the DNA in the specimens and so forth, to both document and try and understand how species respond to environmental change, of which one type is climate change. And of course, climate change is in the news a lot. Whether it's anthropogenic or natural or a combination of them is not my topic tonight. But there is no doubt that climate is changing. Climate has changed in the past. It's changing again. That's the norm. But perhaps now it's changing a little faster than has previously been the case. And many of you will have seen this so-called hockey stick graph. This is going back to about when our study starts in 1900. A bit of bumping around off the instrumental record here. And then a variety of projections into the future depending on how we as a society behave and intersecting natural processes. The sad part is at the moment we're on the worst case trajectory of the IPCC. And a lot of people have put a lot of effort into trying to think about what's the rate of change, the magnitude of change, how does that compare to the past, which parts of the planet will have totally novel climates, that is, climatic conditions such as here in the Amazon are forecasted to be conditions that currently exist nowhere else on the planet. Right, that's the idea of a novel climate. Disappearing climates, such as in the Andes here, areas that where the climate space, the type of rainfall or precipitation that's there now will not exist in the future anywhere on the planet. And this is all done, of course, with a variety of scenarios. And there's a lot of sort of thinking about the biological consequences of this. So there's a large industry based on trying to predict what might happen. This is, I like this, it's kind of colourful. It's also a different place on the planet I work, the rainforests of northeast Queensland. Cairns is about here, if anyone's ever been there. This is a mountain system, and the green to brown here represents the richness, that's the number of rainforest specialised species that live in that place. So the greener, the richer, the more species. And these are different types of models of what might happen as temperature shown on this axis here increases over time. And these are mountains, tropical mountains, all with very unique species. And if you watch the little thing go up, I mean, it's no great surprise, right, that as it gets warmer, I'll show you that again. It's kind of depressing, really. It's better to play it backwards. It looks better. But, you know, this is this sort of idea that as temperatures increase, in general, things will move up. Right? That's very logical. And, of course, as I'll say more about later, changes in rainfall, which are much less certain than changes in temperature, are likely to have lots of effects as well. So there's a large industry now of academics and people in government agencies and NGOs that are trying to forecast, where a species going to go across the planet and across landscapes, given different forecasts of climate change. This has profound consequences, not just for species diversity, but perhaps ecosystem services and maintaining healthy ecological communities. But it's very hard to predict. As you'll see later, the uncertainty around this is very large. So where the museums come in, we learn from the past. Right? So if we have good records from, say, 100 years ago, we can go and see how things have changed over the past 100 years, then obviously we use history to inform the future. That's the mantra in humanities, politics, economics, and it is also in this type of biology. And museums have this unique record. It doesn't exist anywhere else. It's in museums. And museum science is being transformed by new technologies that enable us to ask new questions about these specimens that we've been protecting for future generations as time goes on. So here's our traditional focus, keep mixing the button, on specimens, the objects themselves, the stuffed rats, the stuffed birds, the reptiles in formalin, the pinned insects, the herbarium specimens, all that stuff. As I'll show you now, we can now intersect this knowledge, these specimens, with other types of knowledge that are coming from, say, human biology, the techniques in human genomics, sort of trying to sequence human genomes for disease purposes, and so on has spawned a huge technology that we can apply to a rat or to a mouse or to a plant. Environmental data, remote sensing data, climatic data in time and space, again, is a huge growth area of information. So we can try and knit all these types of information together to get a richer view of how environmental change is combining with changes in specimens, in phenotypes, that's the traits of the specimens, or with the genomes of these specimens. And armed with that, perhaps we can make better predictions or have a better understanding of how things change. So I'm in a very fortunate position, have been for the past, decade as a director of this institution. It's about a bit over 100 years old now. And it was founded by this lady, Annie Alexander, who was an avid hunter and collector. Her father used to take her hunting in Africa. Sadly, on one of those trips, her father, who was the founder of C&H Sugar, very, very wealthy man, he died on one of those trips, fell off a waterfall. Students, be careful on waterfalls if you're pointing a gun. She came back filthy rich and wondering what to do with her money, and she loved the outdoors. She liked science. California Academy of Sciences had just burnt down in the 1906 earthquake. Had been totally destroyed, lost all its collections. And she decided to set up a museum at the in the just developing University of California, and this was the Museum of Vertebrate Zoology. And then she handpicked the director. So this is what donors can't do now. She wrote to the president and said, I'm going to give you this money. I'm going to run the finances. I'm going to pick the director and get out of my way. And the president said, yes, ma'am. Your wish is our command. Formidable, wonderful person. She picked Joe Grinnell, who at the time was a student at Throop Polytechnic, now called Caltech. He was in the first class at Throop Polytechnic, and turned out to be a very influential in development of both ecological and evolutionary theory. He came up with the concept of ecological niches through his field surveys and studies of species replacements. But he also wrote this, and it's a whole bunch of words, and I know we'd say never put too many words on a slide, but I'll let you just sort of scan it while I talk. Soon after I got to the museum, I was talking to my good friend Jim Patton about what's something we can do as sort of a collective effort in the museum to sort of bring us together and really show the value of the collections. And he gave me some things to read, and one of them was this paper written in 1910 called The Uses of a Museum. in which Grinnell basically says that he was establishing this collection with rich backing information so that people could come back in about a century and see what has changed. It was quite prophetic. I got very excited. I read this and said, Jim, look what I found. I showed him this paragraph and he said, yep, that's good. Took me to the library, our library in the museum, and there's a big plaque with these same words on it. So there are very few new ideas, you know. But then I was also struck just recently, we had our open day. We're a research museum, not a public museum. We have an open day. And I was listening to one of our undergraduates talk to a student, and you may not be able to see this, those of you up the back. She was trying to explain what we do and said, you know, a specimen's like a good book. You can read it again and again. I thought that was great coming from one of our undergrads. And I'm going to show you today how we read these books. So, going back to Grinnell's time, it was 1908, a little bit earlier, up to about 1939 when he died. He and his team, funded by Annie Alexander, NSF didn't exist, made these collections, all these spots around California here along the coast. Here's the Sierra Nevada, up and over the Sierra Nevada, in the Mojave and the Colorado deserts down here. Very substantial collections of birds and mammals in particular. And he was interested in how species replace each other across elevational gradients, from which came this idea of ecological niches, and north to south. But he established this fabulous record, and E. Raymond Hall was out working in Nevada a few years later, and right across the Great Basin into Utah. So at the time, Grinnell was quite concerned about the ecological damage already being done to California. So it was not a pristine place. Right? John Muir, the famous environmentalist, had been up in the High Sierra running sheep in the High Sierra, as they do in the Wasatch, used to do in the Wasatch Range. Overgrazing, a lot of damage. The gold mining had used placer mining, big jets of water to blast out the gold. All that sediment ran down to San Francisco Bay, amazingly. And there's intense agricultural development already in the Central Valley, and the cities were expanding. So he could already see California was changing fast and wanted to get this record before conditions were irrevocably altered. This is the current ecological condition of California. Brown is bad, green is good. You can see the obvious things, the agricultural replacement of natural systems, lovely LA Basin, Bay Area and so forth. And then also since the early 20th century, there's been a good deal of climate change, which varies around the state depending on where you are. And this is actually quite hard to put together because the very early climate data is not as extensive, obviously, as the more recent. But blue is, the top one here is minimum temperature, the sort of nighttime temperatures, and red is warming. Particularly you can see the Mojave here is cooking, as are some parts of the southern Great Basin, and some parts of the Sierra. We'll come back to that. And rainfall is changing as well. So there has, over that 100-year period, been some quite substantial change. So what is this? What did Grinnell leave us? Tens of thousands of specimens. The current collection is about 750,000 specimens. Oddly enough, we don't have fish. And the reason for that, so we're a museum of vertebrate zoology sans fish. It sounds like we're pretty poor zoologists if we don't get that. But at the time Grinnell was establishing the museum, there's a guy called David Starr Jordan, who was the eminent ichthyologist in the West. At Stanford, it's this Stanford Berkeley thing that keeps going. He went on to become the president of Stanford, but he had the fish collection. Stanford then gave up all their collections, and along with it, a lot of their organismal biology. But fortunately, that fish collection, David Starr Jordan's fish collection, went to California Academy, where it still is today. Anyway, so sans fish, we have about 750,000 specimens, well over 100,000 originally collected by Grinnell and his colleagues. But aside from the specimens all sitting in these cases, and here's Grinnell at his desk, we still have the desk, we don't throw away anything. There was well over 100,000 pages of field notes. And these are written every day It was precisely what you did, what you caught, what you saw, what you thought about, sometimes what you ate, jokes people told at the campfire that were good. All the stuff gets written down. And we still teach students the writing of field notes in our courses, vertebrate natural history. It's just a skill of being a field biologist, to take good notes. And Grinnell established this system that went beyond the norm. They had maps in there of exactly where their traps was. This is in Lyell Canyon in Yosemite Park. They had daily bird counts. They would walk a particular transect each morning, and every half hour they wrote down the numbers of all the different species they saw. This is pretty routine now. It's called a line transect, but in the early 20th century, it was pretty much unheard of. And lots of photographs. We got well over 10,000 photographs from that period. Here's Lake Vogelssang then, here it is now. You can see the increased stature of the lodgepole pines. But up on these hills, you can see some of the same bushes 100 years ago and now. So this sort of background information makes every specimen so much more valuable, because you understand more about how it was captured and under what environmental conditions. So again, here's the map. Here's some more examples of field notes with these are the specimens, the field number, a bit of morphological data, what it is. There's a gopher, a chipmunk, a Perignathus mouse. Here's the trapping conditions, a bit about the habitat, and so on. So part of our work, we've had a lot of undergrads involved in this project, has been going through these field notes, reading about the history and extracting data from them that we can then use in analyses. So this, what's become called now the Grinnell Resurvey Project, actually has very simple aims. It's to see what's changed and trying to understand why. So we're interested in providing another baseline. So Grinnell's work in the early 20th century gives us one baseline of the ecological conditions. We want to do that again at the same level of detail so people can come back again in 50 years. And now we've got 2 points rather than 1 point. If we can observe changes not just in species distributions, but in community structure, genetic diversity, and so on, try and understand those changes, and then that gives us, we hope, more information to be able to make robust forecasts of which species are impacted, how are biological processes changing, ecologically, evolutionarily, or whatever. And this work has involved a very large number of people. I'm just the ringleader, the person who's really, really driven it and been the inspiration for it is this guy, Jim Patton, who's, what did you call him before? The man. He's the guy, probably the preeminent mammalogist in North America. Steve Beising is the ecological statistician and birder, and then lots of students making things happen over time. And this sort of collective exercise, and I know it's done with classes here as well, is fabulous for undergraduate training, because the undergraduates see the project evolve, they're involved in the field, they're involved in the lab analysing the specimens that come back, doing the curation, they're involved in outreach, explaining the project to other people. So everything from go to woe involves undergraduate students, many of whom stay with us for their whole time on campus. And obviously go on to grad school and so on. So I want to show you a little bit of data to explain the patterns we're seeing, and I hope it's not too heavy. But this is where we did the first of 3 major transects in Yosemite. So here's the Central Valley down here at very low elevation, coming up to about 10,000 feet at the crest here. This is what we call the transect. Each of these little dots are the historic sampling sites that we've been back to resample the fauna. Collecting specimens again, trapping just the way they trapped. The bird methods are slightly different but comparable. So here's the outline of Yosemite National Park, the crest here dropping over into Lake Mono and the desert on the east side. So when these guys did it, they produced this monograph that's still the basic reference for knowledge of the vertebrates of the Sierra Nevada. In fact, the National Park Service have just digitized this and put it online because it's so valuable to their people. So they did this over about 6 years, 1914 up to 1920, but most of it was 1915 to 1917. It was a better place to be than the Western Front, for sure. And involved about 1,000 man days in the field, and they were Grinnell's words. So this was all funded by a woman, Annie Alexander. But in those days, men and women did not go in the field together. So Annie and her lifelong colleague, Louise Kellogg, were out trapping in other places, doing other things, and the guys were all out here in the field in Yosemite doing this work. So 3,000 pages of field notes, almost 4,500 specimens, 700 photographs, just from this one little area here. A super rich record. So I'll just show you a couple of cartoon versions of some of the changes we've seen over this sort of 100 or 80, 90-year period, and then I'll show you some summary graphs just so you know what I'm saying is actually supported by evidence. So here's a sort of profile going from this Central Valley up over the crest to Mono Lake on this side. And these little sort of cartoon boxes are the elevational range in light pink in Grinnell's time and then in the present. So this is a Pinion Mouse, which is sort of well known as a sort of weedy species. Its populations fluctuate a lot over time. And on the east, on the Sierra slope, the west side, it's sort of translocated up by about 500 meters. So the bottom limit has moved up, the top limit has moved up. It's just sort of done that going up the mountain. Okay? Amazingly, on the east side, we now have it colonizing the habitats up here. It's all quite separated from these. And through genetics and morphology, we're able to show that the mice found here now didn't come from here, they came from over here. And they've moved up almost 1,000 meters over 100 years. That's a really big shift. Of course, it's on the X scale, it's not that far because the thing here is so steep. But more importantly, it's shifted its habitat. This thing is called a Pinion Mouse. Duke Rogers reminded me earlier today that they see it in lots of habitats other than Pinion in the Great Basin. But now it's up in montane conifer forest, hemlock and whitebark pine. So it's shifted its ecological range quite dramatically over 100 years, and it sort of reminds us that a lot of what we think we know about species is sort of a snapshot in time. We've only observed them for a few decades with modern science. And as environments change, species can surprise us. You get a lot of ecological surprises. So this little mouse, cute little guy, big long ears, is doing beautifully well. We're seeing a lot of higher elevation species that are not having such a good time. This is the golden-mantled squirrel, really beautiful animal, also in the Sky Islands around the Great Basin. Its lower limit has contracted up about the same distance, about 500 feet, and it really can't go any higher because it runs out of land. Right. This data actually is a bit dated now. This is the pika. There's been a lot of concern about that. People are really concerned about the pika because it's so cute, sort of the polar bear of the Sierras. There's photos of it with flowers between its teeth, looking like in fact, further surveys have suggested the pika is actually not in much trouble in the Sierra Nevada. We got a small change, more sampling, improved that result. Now, because it's real data, not everything moves up, which is what we expect with warming, right? So the shrews are doing all sorts of crazy stuff. This shrew, which is a wet meadow specialist, has moved down by about 1,000 feet compared to its previous history. We think that might be because one thing the park has been working very hard on is restoring wet meadows, restoring the hydrology of the meadow systems, which were, in the early 20th century, were badly impacted by overgrazing from sheep. Sheep are excluded, the wet meadows are improving, and these wet meadow specialist species are expanding. So the good news is over this sort of 100-year period, Yosemite hasn't lost species. The grizzly was already gone in Grinnell's time. Wolverines were exceptionally rare. I only saw one, I think, or two. Porcupines are gone. They were hunted out. But overall, for small mammals, there's really been no loss of species richness. These bars are how many species are found in the park as a whole. The black is in the past, grey is now. There's really no change. There's a bit of a drop at the lower elevations outside of the park. But overall, the park is doing its job. Species are moving back and forth, but because they can migrate, you're not having species extinctions. So national parks, big national parks across environmental gradients are good. They're doing their job. Now I'm going to show you a few bar graphs like this, so bear with me because I want you to see the scale of the data that we can now get. I can barely see it myself from here. So on this axis you've got the elevation, so right from sea level up to about 4,000 metres. And each bar here is a different species. So there's 28 species across here. Green means it's a significant expansion of the range. Red means it's a contraction of the range. So take this one, Alpinus. It used to occur down to about 2,500 metres. Its bottom limit is now 3,000 metres. Okay, so red, and these are all highly significant statistically. I can explain to number junkies later how we do that. But these are the results we're very confident in. We're very conservative in how we do it. So out here we've got a bunch of species that have not changed their elevational range over 100 years. Fine. Over here we've got mostly low elevation species that have expanded their ranges, mostly up. This is that shrew I showed you that's moved down. But mostly low elevation things moving up, sometimes up from the bottom and expanding up from the top. And then these are the high elevation species, some of which are undergoing range collapse. This is the bushy-tailed wood rat, Neotoma cinerea. It used to be over this range, very common. Now it's only found in a very small place. This is Allen chipmunk. Again, it's had sort of a range collapse. Then others are sort of being pushed up. And these are not subtle changes. The average elevational change here for species that have shifted is 500 metres. Just in elevation, right? That's a long way up. So we feel like these are pretty strong results. The other thing to note is there's a lot of variation among very similar species here. All the ones starting with T here, this one, and this one, they're all chipmunks. So we have some species collapsing, some moving up, some not changing at all. So the local communities, the local mix of any one place on the landscape, the types of chipmunks that is present is changing. The community's dynamics is changing a lot. And this is consistent with what we see in the fossil record. When you study the fossil record through time, communities don't move as one piece. They move in pieces and they get reshuffled with climate change in the past. And this is the same thing we're seeing now, but on a much shorter time scale. So what's going on? You probably have a pretty good idea already. The dominant signature here is of things moving up, either expanding upwards if you're previously a low elevation species, or contracting up if you're a species that already went to the mountaintop. And that smacks of warming. That's exactly what we'd expect. So this is the instrumental record, the 100-year weather record from Yosemite Valley. These are nighttime temperatures, not average, but nighttime temperatures, and by different seasons. And a conservative estimate is that the average nighttime temperature has gone up by about 3 degrees Celsius in 100 years. That's a lot more than the average across all of California, which is a bit less than 1 degree. But the Central Sierra, for some reason, has warmed up quite dramatically. And we see it also in the contracting glaciers. This is Lyell Glacier at about that time, 1903. This is it now. And there's other data from tree rings and expansion of pines into meadows, consistent with quite strong warming in this part of California. Now, that's not the only thing going on. So before we jump to the conclusion that this is all due to warming, we also need to consider habitat change, because we've also changed the way we manage the forests. And a lot of you are familiar with fire suppression. So there was a long-term policy over the last decade of stopping fires. So the fuel load increases, the forests get denser, red fir forests in particular. So you've got a lot of habitat change going on, largely unrelated to climate change. So you've got to sort of try and understand, could that explain everything we're seeing? We're lucky, again, in this system, to have vegetation maps from the 1920s and 1930s. This was done by a professor at Berkeley called Wieslander, who did the original vegetation mapping for California. And these maps were very nearly thrown out when he, I think, died. Anyway, for some reason vacating his office. Death is one way to do that. Someone else was cleaning it out and throwing all this stuff out of his filing cabinets into the big wheelie bins. And another professor happened to wander past and said, those maps might be useful to someone. And these was the only copy of these vegetation maps of California from the 1920s. So they were rescued, they're now digitized, and we can compare directly the distribution of vegetation types. This is the Yosemite Transect from Central Valley over to Mono Lake. We can compare directly these vegetation maps and ask, can we explain the changes in species range by the changes in the range of their habitats? And I won't go through the data, just to see we have data. The answer is for the species that are expanding, it does seem to be connected to the vegetation change. A lot of these are species that react very strongly to fire. So as you go from dense conifer to more open habitats, these species colonize. That's natural, that's normal. We can't explain the high elevation species. All these, so the stuff up in here fits the model, stuff down here fits the model. These ones don't. So a lot of these species, the red dots are the species, that the high elevation species that are contracting. We wouldn't have expected that based on the vegetation. So it really does seem to be a direct effect of physiology, of something to do with climate. Okay. So that was our first pass of this in Yosemite. That's just one place, right? So we've been expanding these studies either for a particular species, trying to get a more geographic extensive view of how it's changed, or for the whole community. So this is one of these examples of the species level studies. It's for Belden's Ground School, a really cute little guy. Sometimes called the picket pin. They sit up like this in the meadows and give alarm calls when you come past. Really cute little squirrels. They've disappeared from 45% of their historic localities, mostly at lower, warmer elevations. We've not seen any natural colonization of places they weren't previously present. So the species seems to be undergoing a pretty strong decline, at least in this part of its geographic range. The same species goes right up into Canada, down here through into the Rockies. We don't know what's happening there. But at least in the Sierra Nevada, it seems to be disappearing, largely from the warmer parts of the range. And we have lots of students now studying that. This was the Yosemite data here. This is just the species, the high elevation species. Here's the Yosemite Transect. We've done transects now up at Lassen and down in the southern Sierras. The pattern of climate change is a bit different in these different transects. So if you've got arrows coming on in this direction, it means it's warming. It's going up that way, it's getting wetter. It's getting wetter across most of the Sierra Nevada. And in particular, it's getting notably warmer in Yosemite and in the southern Sierra. And the general pattern that emerges from all of these with statistical analysis is the most consistent feature isn't the low elevation ones expanding. That's very hit and miss as you go across these different transects. But what is consistent is the high elevation species contracting upwards from the bottom of their distribution. That's a very repeatable pattern across these different transects. So we do seem to be seeing a very general result that high elevation species are contracting. Now, this is the same sort of data for birds. There's a lot more birds out there that we can survey. Morgan Tingley has gone to the same places, following the footsteps again of Grinnell and so forth, using these field notes to know exactly what they saw and when, redoing the surveys in the same places, again in the Lassen, Yosemite, the southern Sierra. And this is much more heterogeneous than the mammals. There isn't really any strong overall signal of things moving up or moving down. It's a real hodgepodge. It just looks like a sort of a dynamic community fluctuating back and forth across the landscape. So here our sort of naive view that it's getting warmer, particularly in these places. We ought to see most things moving up, is not being met by the birds. They're doing something very different to the mammals. And we've been thinking about this a lot. And and trying different ways to understand it. Again, I'm sorry, this is a very data-intense slide. Of course, it's not just temperature that's changing. Precipitation is changing. So the average rainfall is going up. Less of it's coming as snow, more of it's coming as rain, with increasing nighttime temperatures. In some places, the dry season rainfall is less. So you're getting more rainfall overall, but the seasonality is changing. That's going to affect the vegetation, the primary productivity that drives the insects that the birds eat, and so on. So we've been trying to now look at these two factors separately. And just to take one example here for Yosemite, this is the temperature change, but this is now taking the range limit for each species, and red means that you would predict by temperature it would move up, all of these. So if temperature alone is driving it, it should move up. If the species is actually determined more by rainfall, by precipitation, in these same places it should move down. The direction of change in temperature and rainfall is not the same. It can be opposing in terms of which way you would move to stay in the same climate conditions. So a lot of this is going to come down to are species more limited by precipitation or by temperature or by an interaction between the two. So most of what we read about and think about is just things moving up with warming. There's a lot more going on apart from warming. And we do get a better explanation of the results. All of these species are better explained if we consider both temperature and rainfall rather than just temperature. But we have a long way to go in this. Now, before I scare the socks off you with that. So it is complicated for the birds. For the mammals, we've got a pretty consistent uphill move going on, particularly the high elevation species contracting. But there is a tremendous amount of heterogeneity. Even close related species like those different chipmunks I showed you are responding in different ways, resulting in community change or change in community composition. So far, we've tried to do lots of analyses saying, can we correlate whether species respond or not with their life history, with their reproductive cycle, with do they hibernate or not, with their body size, with do they eat insects, do they eat seeds, And we can't find any consistent predictor based on the sort of usual biological attributes. So we know there's a lot of variation between species. We can't predict a priori what types of species will be more sensitive than others, at least not yet. So this is sort of now, like most types of science, you get part of the answer and then it spawns a whole lot of new questions. And this is where we're at. So on the right here, this horrifying diagram with boxes and arrows and colours. This is one group of scientists, Steve Williams and co.'s view on the things you really need to understand to predict species vulnerability to climate change. There's a lot of boxes there. Don't even try and read it. Some of these are things that are properties of the species. Some are property of the habitat. Some are ecological. Some are evolutionary. And we can't fill in those boxes for any organism on Earth, I would suggest. Nonetheless, you can pick off pieces of it and try and understand it. So, but basically it comes down to, is the species exposed to climate change? Is climate change happening where it is? Is it for some reason vulnerable to those changes? And then if it is vulnerable, then you can predict some sort of impact. So the exposure you can measure just by looking at how climate is changing. Vulnerability is all these boxes that you've got to try and figure out. It's a daunting task. But if we want to really understand how species are changing, then we need to do more research. The answer is always more research, otherwise we'd be out of a job. So what we can do with the system now is we can pick off some pairs of species, closely related species, some of which are showing dramatic and consistent change, and some of which are not changing at all, and ask questions about their ecology, their so-called autocology, their ecology, their physiology, their genetics, their phenotypes, and see if we can get a better understanding. So we now have a bunch of students looking at different dimensions. This is one of our favourites, the Lodgepole Chipmunk. This is the historic distribution, again with elevation on this axis. This is the current, no change. And this is what's happened to the Alpine Chipmunk. As I showed you before, it's lost a whole chunk of its lower range. And this is of some concern. This chipmunk is only found in the High Sierra. If it disappears from there, it's gone from the planet. Speciosis, which is not so badly impacted, is also a Sierra endemic. So, what else can we do with specimens? Well, we can look at morphology. So we've got the skulls, we've got the skins from 100 years ago and now, and Grinnell took big samples. He didn't just take one from a meadow, he'd take 20 or 30. Sort of carnage in a way, but now in fact we see the value. We have population sample that we can do statistics on and really do rigorous science. So we've had students look at morphology, and here's the one representation of the historic morphology. This axis here is size of the skull, and this species has increased in size quite dramatically from the historic time to the present. So its range is contracting, yet it's getting bigger. And it's not getting bigger overall. We have a student from Brazil working with us now, Ana Paula Assis and Gabriel Maroig, who are doing the very detailed study of morphometrics, measuring lots of different points on the skull, trying to find out where is that skull getting bigger and how does that relate to the functions that are implied by the skull. And what we're seeing is that this part of the skull this part's not changing. This part of the skull is extending, the rostrum. And also this tooth is changing in direction as well. But this is the big change. And one possibility is there, this is where the nasal membranes sit. I'm a lizard guy, by the way. I'm not a mammalogist, so forgive me, Duke, if I get it wrong. Where are you, Duke? You there? Help. Right. So the idea is this is getting longer. You've got more nasal membranes. You're better able to retain moisture. How did I do? Did I pass? I was taught by Jim, So perhaps it's something to do with drying in the late dry season. We don't know, but it sets up some sort of mechanistic hypothesis that might explain what's going on. Now, curiously, as I said, that's one pair of species. We can look also at the ground squirrels. This cute little Belding's chipmunk, lateralis, which is showing a slight decline in some other areas, not in Yosemite but in other places, against the California ground squirrel, which is not changing its range at all. And amazingly, we see the same thing. This is skull length here on this axis. The species that are contracting their range are getting bigger again. That's a real surprise to me. For some reason, I thought species are losing their range, they must be getting smaller. But they're not, they're getting bigger, and getting bigger in interesting ways. And this is work that's just got a long way to go. It's really interesting. And this is not just a consequence of it's lost the low elevation ones. It's independent of elevational range, but the contracting species are getting bigger in skull size. I don't know what it means, but someone brighter than me might figure that out. Another thing we can do with the skins, we can pull off hair samples and look at what are called stable isotopes. So things like carbon and nitrogen come in different isotopes, different numbers of neutrons in the nucleus. And we know a lot about how the ratios of those different forms, say of nitrogen on this axis or carbon on this axis, relate to climate and also to vegetation. So in climate, you go from C3 to C4 grasses, more arid out this side, wetter over on this side. If you've got nitrogen-fixing plants in the diet, you'll be down this part of the axis if they're eating more in the forest and not with nitrogen-fixing plants that are up there. So we can look at the historic specimens from 100 years ago and do these chemical analyses of the isotope ratios and do the same thing with the modern specimens. Try and get some clues on changes in diet over 100 years. And again, we're seeing something interesting that we're still trying to interpret. So here's our sort of comparison again. Here's the species that not have any real consistent change in elevational range. There's lots of change, but it's all over the place. It's sort of random. Or that's one of my Californian words. I've got to say it, random. It's like whatever. That's the only two words I've learned in 10 years in California. So this is random. And this is whatever. No, you can see this. There is a sort of directionality to this, right? So they do seem to be, the hypothesis is that they're eating more legumes. Now, how that relates to changes in geographic range, we don't know. But we're just trying to find, given that all these other correlations we ran failed, didn't tell us anything, we're trying to find the smoking gun here. What is it that's mechanistically affecting these species? The bit that I do know something about, because because I'm actually a geneticist by training, is trying to use genetics to try and understand a number of things. So one is, as geographic ranges get smaller, are the individuals all just moving up the hill, or are we having local extinctions? Having local extinctions, you should have less genetic diversity now than you had in the past. Populations are smaller. If the geographic range is fragmented, you should see more genetic differences between populations than you did in the past. And the holy grail is to be able to find genes or metabolic pathways in which the genes are under strong selection. And that might point us back to the phenotype, to the sort of organismal trait that might be strongly influencing whether species persist or decline. So these skins, these beautiful little chipmunk skins with associated skulls, are little bags of DNA. But the DNA is really chopped up, so it's very fragmented just by random chemical damage from sitting in a drawer over 100 years. So in the past, this has been a challenge. Some people have done it, but as I'll show you, we're on the cusp of something really exciting. So one of our students, Emily Rumage, did a heroic study using microsatellites. These are genes that have lots of variation but are very short gene products. So even if the DNA is fragmented, you can often assay them, but it's really laborious. But she did that. And for the alpine chipmunk, our sort of canary in the system, here's a portrayal of the original geographic extent. It's a model based on elevation. You can see big continuous population along the Sierra Crest. This is what we think it is now, much more fragmented, much smaller geographic range. So what we expect to see again was a reduce in diversity overall and increases differences among populations, and that's exactly what she saw. Here's the historic thing off the skins from 100 years ago, that in terms of genetic diversity, it's dropped to the present with our new samples. And this is a measure of the difference or the isolation among populations. In the past, effectively zero. Now it's quite substantial. So using the genetics here, we've been able to, and from these skins, using methods that were unimaginable in Grinnell's day, we're getting a new type of information that he wouldn't have imagined. And in the species that hasn't changed its range, there's no change in the genetics. So that's fun. It was hard work. What we can do now is really awesome. So for the Human Genome Project, you've probably heard about this idea of being able to sequence a genome for $1,000 or $10,000, personalized medicine, all this stuff. The technology for that sequences DNA across the whole genome, but in very short pieces, like 100 nucleotides long, and then pieces all that together bioinformatically. The DNA in the skins is ideal for this because it's already in little tiny pieces. So we don't, if you take modern DNA, you've got to cut it into those little tiny pieces. You've got to shear it, chop it up like crazy before you sequence it. The skins are already there. It's so cool. So these are the sequencing libraries prepared of 20 skins. This is a measure of the sequencing success. These are the skins. These are the modern samples. There's some other species of chipmunk. In fact, the 100-year-old skins provide better sequence data than the modern stuff. Now, there's some issues with DNA damage that we're dealing with now, but we did this for 12,000 genes in one experiment. It took Emily 2.5 years of her PhD to do six genes. So this is the transformation that modern genomics is taking us to. And again, we can only do this because we've protected the skins over this time. So the notion is that we can take these examples where species have contracted their range, as alpinus. It's happened in 2 places. We have the samples. We expect to see strong selection here, but not in our control, the species that hasn't changed its range. And so we take all this wonderful data, we combine it with a guy with a really big brain called Rasmus Nielsen, who's been doing a lot of the human genetics, and hopefully we get answers that illuminate what's going on. So, this is the first pass. This work has taken us 5 or 6 years now. And, this POX plot here is the distribution of specimens of birds and mammals in California, but then coming out into the Great Basin of Mojave. We're over here somewhere. The Great Basin has a very rich historical record, both in our museum and the Utah Museum of Natural History, and at Kansas. And the group from the Utah Museum, here, Eric Ruckheit and his colleagues, have been out doing the same thing, resurveying these historical sites in the Toyabes, Rubies, and so on. Red here means there's been more warming since the early 20th century. And as I said before, the Mojave is cooking. Great Basin hasn't warmed anywhere near as much, at least by this measure, whereas areas like Joshua Tree, Mojave National Monument, Death Valley have got a much, much warmer over this time. So these areas also would be fabulous to resurvey. So a lot of this stuff is coming together at Berkeley in a new initiative called the Global Change Biology Initiative. We're trying to bring together the geneticists and the ecologists and the museum people and the genomics types and bioinformaticians to tackle some of these problems. And I'd like to think that when people come back in another 1,500 years, they'll be doing things not just with Grinnell specimens, but the ones we've just collected again, and asking questions and using techniques that to us now are unimaginable. The key thing is protecting the collections, such as the one you have here. Thank you. And of course, I'm very happy to take questions if there are any. Yes?
So you talked about changes in presence, absence, changes in range. What about abundance?
We'd love to be able to do that. It's much harder. The historical record is good, but they weren't as systematic in the way they observed the birds or trapped the mammals as we are now. So we can look at relative abundance. There's some very interesting patterns coming out of that, but we can't look at absolute abundance. That's just too difficult to do with a historic record. But we're now very careful to do things in a way that will yield abundance estimates when people come back again in the future. Very good question. Other questions? Someone? Yes.
Through my years and working with wildlife management, it seems like you've concentrated more on the habitat. There's nothing in the or put in the studies about predation on these animals.
Mm-hmm.
And is that the set predation? Does it have any effect?
Mm.
Is it ignored?
Mm.
What
Very good question. So for those in here up the back, the question is, what's happened with the predators? Has the pattern of predation changed over time?
not only predators, but there's disease.
Yes.
There's big bugs that eat little bugs,
Mm-hmm.
and so on.
You're right. So this is this whole area of biotic interactions. Yeah.
Like in ecology, it's not just one thing. It's a slice of a whole lot of pie.
Mm-hmm.
And if you get the true picture, you've got to take something from each one of those sciences and put it together. And I'm just getting one viewpoint on climate.
Right.
Well, take it from there.
Yep, no, absolutely. So I guess the criticism, and it's a very valid one, is we're focusing so much on climate, we're ignoring all the other biological processes, biological interactions that are going on. And that's a very valid point. It's really hard to know how things like predation, disease, parasitism, and so forth is affecting these, because that's not something we can really address using the 100-year-old record versus now. We do know that predators in the system have changed. So some of the Fisher and Martin, the specimens upstairs in the displays, the sort of mesopredators are nowhere near as common as they were previously. Wolverines are gone. But there's still lots of coyotes and so forth running around there, and as well as weasels and the smaller predators. So no doubt there are changes in biological interactions. I don't think they would necessarily explain this more consistent pattern of upward shifts of elevational ranges in the high elevation species. But if I could figure out a way to test that and exclude biological interactions, we are going to try and look at disease. There are some diseases that we can detect in the skins, and I've been talking to Duke about this today. So Lyme disease, perhaps plague, we can probably get out of the teeth, remnants of plague DNA or antibodies. Duke is more of an expert on this than I am. So most of these other things I think would have an effect, but not such a consistent effect, or not. You wouldn't get this overall pattern of the high elevation species contracting upwards. I think it probably would explain, if we could understand, a lot of the heterogeneity among species. The fact that, particularly birds are all over the place, and there's a lot of, in between closely related species, there's a lot of very different things happening. And I think a lot of that will have to do with these other elements that we simply haven't figured out how to address on this time span. I don't know if that answers your question, but it's a daunting challenge.
I don't hear very well, but what I heard doesn't answer my question.
That's right. Okay, so you're very possibly right, but it's very hard to study. I don't think it would generate the overall pattern we see. But if someone could figure out a way to prove me wrong, I'd be delighted. But it may help explain, if we could understand how to look at effects of disease, effects of predators, it might help explain the differences we see among closely related species and how they're responding.
Well, just to follow up, in our excellent wildlife habitat for mule deer and elk and so on, the high elevation summer ranges, elk are doing okay, but the deer, which the predators, cougars, coyotes, so on, prey on, they've left the high country and they've migrated, and they're down in the valleys and the subdivisions along the rivers and so on to get away from the predators. And if you were measuring these other smaller species and so on, that's all that's left for the predators to eat in the high country unless they come down to the lower country and follow the deer. And I see a close relationship between predation and population of species. And there's no information here to answer my question.
No, you're absolutely right. Yep. So what we need to have on that is consistent surveys of the distribution of predators and particularly the cougars and so forth from 100 years ago to now, and we don't have that data.
We do have some of it in studies on predation and prey species and predators. The population of the prey species is high, and the predators go up, prey species go down, and then the predators go down, and so there's a one population curve follows the other.
Yeah, that's well known. Yeah.
We try to balance that out so we're more of a straight line. We don't have feast and famine.
Right.
Right now, for our big game population of deer, we're in a famine.
Yep.
It doesn't look like we're going to get out of it very easy.
Yeah, well, this does fluctuate a lot, the rodent numbers, and I assume the predators, but I don't know, that changes a lot from year to year, depending on snowpack, of course, as well. That's a major driver. Yes, there's a question just behind you. Yeah.
Among the species that have undergone a morphological change, are there common genes or common changes in genes or metabolic pathways among those species?
Yeah, we don't know yet. So that's exactly what we're looking for. So we've now sort of focused our search on these genes, in part on genes that are known in mouse, for example, to be associated with those types of shifts in morphology, morphogenic proteins and so on. So it's a bit of a fishing trip, as you can imagine, but that's a really good question, and come to Berkeley and study it. Other questions? Yes?
Are there intermediate collections through time that could be used to sort of do something about
Yep.
just the 2 points? I'm sure there are some samples available that might
Yeah.
some of those.
Nothing is systematic, because the museum did these general surveys up until the 1940s and a bit into the 50s. And then there was a period where people went out, like Jim, chasing every gopher that was alive in California, but not doing general surveys. So you can't use that sort of data. Now there is, as we've published this stuff, people have been contacting, and we talked to one guy who was a wildlife biologist who worked in the White Mountains and had to go from Bishop up to to the Barcroft lab for doing wildlife surveys regularly, and he was counting the chipmunks as he went up and down that hill at various places for 40 years, from 1960 through to 2000, and he's got all his field notes now at Calicat. That's a treasure trove of data, because with that we can start to address your question of population dynamics through time. So but there's very little data like that. You know, monitoring is really important, but it's really unsexy. It's very hard to get money to do routine monitoring like this. But then you see this sort of data and think, God, I wish I had data from the 1940s, 1950s, 1960s, 1970s. Yes, up the back.
This sort of relates to the idea, too, that kind of what you described is a problem that you encounter is there are just so many variables that there's a limit to how many things you can consider.
Mm-hmm.
And so we can consider rainfall and temperature, but maybe not predators or disease. You know, there are so many different things. Are you aware of any sort of efforts or potential efforts to use computing, to use advances in computing power, to sort of begin to compile more and more and more variables into the same model? I don't know if you understand the question. I didn't phrase it quite right. I mean, is there a trend of being able to look at, say, the data for population based on temperature, population based on other factors, and combining that data?
Yes. So, I mean, you can do that with, multivariate statistics and so forth, AIC-type models where you weight different variables. And it's all correlational. And in fact, I think we need to move away from that. I think we need to come up with very clear hypotheses based on mechanisms, on physiology, on plant productivity and food webs and so forth, and try and really focus our questions much more on sort of mechanistic hypotheses. We've tried the sort of ecological statistical fishing trips and come up empty-handed. So I think we need to really rethink our approach and get the critical data that's necessary, whether it's predators, as that gentleman mentioned, or physiological parameters. We know a lot about the physiology of these animals. They were studied intensively in the 50s, 60s, and 70s. I'm a lizard genetics guy. We need to collect. This stuff is really cool collaborative science. We need high-end statisticians, computer scientists, physiologists, ecologists, plant biologists working with us to understand what's going on. Other questions? Well, if not, thank you very much.